Hello, everyone, and welcome to today's live broadcast, Beyond Sequencing, Population Analysis in an Era of Mass Spectrometry Proteomics, presented by Dr. Evan Williams. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Today's webinar is sponsored by SCIEX. SCIEX's global leadership and world-class service and support in the capillary, electrophoresis, and liquid chromatography mass spectrometry industry have made it a trusted partner to thousands of scientists and lab analysts worldwide who are focused on the basic research, drug discovery and development, food and environmental testing, forensics, and clinical research. For more information, please go to www.sciex.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We will answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window. Or you can use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce our speaker, Dr. Evan Williams. Evan Williams obtained his bachelor's degree in bioengineering at Rice University and his doctorate in bioengineering at the EPFL in Switzerland. At the EPFL, Evan examined a genetically diverse mouse population to identify the consequences of different diets and genetic backgrounds on the development of metabolic diseases and traits. His doctoral research concerned a systems biology approach to combining multiple omics datasets, genomics, transcriptomics, proteomics, and metabolomics, to identify novel gene phenotype links. This project identified and validated novel gene targets, including for diabetes susceptibility and for varying mitochondrial activity. Since 2016, Evan has worked with the group Professor Rudi Aebersold at the ETH Zurich, where he currently works on implementing and expanding the capacity of SWATH-MS proteomics for population analysis. Evan's primary projects now focus on expanding the capacity and accuracy of proteomics measurements to examine hypotheses that are uniquely apparent from proteomic measurements. Please join me in welcoming Dr. Evan Williams. I will now turn the presentation over to him. Dr. Williams?
Hi, thanks, Christy. So again, as she just introduced me, I'm a scientist at the ETH Zurich in Rudi Aebersold's group, who has spent several decades now designing new technologies for mass spectrometry-based proteomics. So I'm a geneticist by training, and I've been collaborating with Rudi's group for several years now on how to integrate these next-gen systems proteomics techniques together with genomics, transcriptomics, and metabolomics in order to identify how natural genetic variants across a population, how these interact to modify our phenotypes, particularly looking at our metabolic phenotypes and our mitochondria. So this talk, however, will primarily focus on the recent proteomic developments in history and how this can be applied by scientists who are not themselves specialists in mass spectrometry to their own biological hypotheses and questions. So the primary two sections of this talk are, first off, to highlight what's new in proteomics and what's old in proteomics and how things have improved and why proteomics is now again becoming one of those technologies that is now very much mainstream and is getting used alongside transcriptomics more and more frequently. So about 10 years ago, you probably heard of proteomics for the first time being generally applied in shotgun proteomics, But back then it sort of fizzled out as it didn't quite deliver what people were hoping for. So people were hoping that proteomics would be the equivalent of transcriptomics in that you get very reliable measurements of the entire proteome of 15 or 20,000 proteins consistently and very quickly and very cheaply. So this didn't quite happen 10 years ago, but now in the past few years, especially the last couple of years, it's become a mainstream technology where you can measure several thousand proteins in single samples reliably and quickly. So now it is becoming a technology that's really going alongside with transcriptomics and genomics and other nucleotide sequencing projects. So in the latter half of this talk, for the implementation and application, I'll bring up a couple examples of studies that actually need proteomics and these applications that you could not do with transcriptomics or DNA sequencing, and these same studies that would be basically impossible by using antibody-based techniques. So it's something that really needs these new developments in mass spectrometers and mass spectrometer applications in order to develop and examine these biological pathways. So the theory of modern mass spectrometry proteomics is divided into two basic concepts. There's the top-down proteomics, which measures the entire level of a protein, but more commonly there's the bottom-up proteomics, which is what we'll discuss here. So bottom-up proteomics, you take your total sample, your protein, and then you fragment it with something like trypsin, and this gives you a whole bunch of short peptide sequences. These peptide sequences you can then shoot through your mass spectrometer, and then eventually you get a mass-to-charge ratio and an intensity, and then these intensities map to particular peptides, and then you can try to associate these peptides back with the proteins that they're originally from. So this is the most common type of mass spectrometry today, and especially if you're doing large scale, if you want to measure dozens or hundreds of thousands, all of these technologies are now using bottom-up proteomics. So that's what we'll focus on here today. So right now there are a tremendous number of mass spec proteomics techniques. If you look at all of the different acronyms for them, SWATH, HRM, Shotgun, AIF, PASF, it's really kind of bewildering perhaps, and it's hard to find good reviews that comprehensively tell you what all of these techniques are. I'll suggest a few different ones here in the bottom right. You can see these PubMed IDs, and it'll give you a place to start, although again, it won't tell you everything. However, one thing to keep in mind is that all of these mass spec bottom-up proteomics techniques fall into the same three broad categories. So I'll go into this a little bit more in the next slide, but these are DDA. This is the most commonly familiar type. This is what shotgun proteomics is. There's targeted proteomics, which is kind of like the equivalent, the mass spec equivalent of using ELISAs. And then there's DIA, which is a newer concept, which is kind of a hybrid halfway between targeted and halfway between DDA. So like targeted proteomics, DIA requires a search space. You have to know which peptides you're looking for. But unlike targeted, the search space for DIA is a bioinformatics search space. It's something that you can predict or develop one time and reuse. It's not a physical or chemical sample that you spike in each time. So between these three concepts and all of these sub-acronym applications, there are a number of trade-offs between them, particularly on coverage, accuracy, speed, and cost. Of course, cost is sort of a subset of speed. And this is a brief oversimplification, but generally you can consider that DDA probably has the largest coverage, targeted is probably the most accurate, and DIA is probably the fastest. Although, of course, this depends on exactly what you're trying to do. So I think targeted proteomics is probably a fairly straightforward concept. The idea is you pick some proteins you're interested in, you design and synthesize the peptides, the heavy peptides corresponding to those you wish to measure, you mix them in with your sample, and then you run the mass spectrometer. So this is sort of low throughput but high accuracy, and if you're only interested in studying a couple hundred proteins in a particular pathway, targeted works very well. However, if you're trying to get a whole proteome, or if you're trying to do hypothesis discovery, in this case you have to go into either DDA or DIA. So DDA, which is called data-dependent acquisition, it doesn't know what peptides it's going to find or what it's going to measure in advance. Essentially for DDA, you run your sample through the mass spectrometer and it picks the mass spectrometry peaks and it tries to find whichever peptides it can out of your sample, which is usually those with the higher abundance. They have the spectral peaks that are the highest and they're the most likely to be selected by the mass spectrometer. There are a couple problems with these with this approach, though, with the DDA, the first problem is, again, what I said, is that it usually picks the most abundant peptides. These are not the most interesting necessarily. And the second problem is that it's different between every sample run. So in this case, what I've shown here, if you run 200 different sample runs, in DDA, what you measure in sample 1 is not necessarily the same proteins that you measure in sample 2. So in this case, if you measure 3,000 or 4,000 proteins across 200 samples, maybe only 500 or 1,000 of these proteins are identified in more than half of your samples. It means that many of these proteins you're identifying are only in a couple of samples here and there because for whatever reason, the mass spectrometer in this one sample selected this particular peptide, whereas in another mass spec run, it selected a different set of peptides. So the main problem with this, and the reason that the coverage is low or that the completion matrix is low, is actually that mass spectrometers weren't fast enough to select and fragment every single ion in every single window. So when you inject a total proteome sample, it's very complex. So there are a few ways around this, and the traditional way around it in shotgun or DDA approaches is you fractionate your samples. So this means rather than putting in one injection of your mass spectrometer that has all of the protein in it, you can actually segregate it into different fractions. So maybe you have, you simplify your sample, you divide it into 12 different fractions like I've shown here, and then you can run all of these 12 different fractions through your mass spectrometer one by one by one by one, and then you can sum up this data later on, and then you get a much larger search space, and your completion matrix is much higher. So the downside of this is if you're running 12 fractions for each sample, this takes you 12 times as long to run. So there are these other technologies that assist in this, particularly TMT, tandem mass technology labeling. So you can run multiple samples at the same time. So although in this case here, if you run 12 fractions by 200 samples, that might take you 6 months to actually run all of your samples through a mass spectrometer, which is prohibitive. Even if you do these tandem mass techniques, you'll still take several weeks or even a couple months if you want to get really good coverage across hundreds of different samples. So one of the solutions that's been developed to this fundamental problem is DIA approaches. So in this case, it's data-independent acquisition techniques, and one of the major ones here is SWATH, which is what we use in our group. So this attempts to bypass this problem of fractionations and very long sample acquisition times by a different way. So here what you do is you fractionate one sample from each of your study groups. So in this case, I would fractionate one liver and one heart, and I would run these on the data-dependent acquisition mode, and this would generate my search space. This would tell me all of the proteins that I should be able to find in this sample. So rather than going theoretically and using the DNA sequence to calculate every possible peptide that could ever exist, now I'm only going to do the ones that are actually in the samples. So of course, this gives you much more accurate data, and it's a much smaller search space than attempting to do it bioinformatically completely. So these libraries, you can either generate them yourselves, or if you're using a common organism, you can download them online from a resource like SWATH Atlas. So once you either generated your library or you downloaded it from online, now with the DIA modes, you no longer need to fractionate your samples. You can simply run them directly in a single run, and now you're going to get pretty good coverage. So you might get 3,000 to 4,000 proteins in a single 1-hour run. So this theoretically doesn't get you as many proteins as you could possibly get with DDA, but it's much, much faster, which eliminates a lot of other problems, particularly the cost. The machine time is expensive. And the other thing is that you get batch effects. There are issues if you run 6 months over time. Your machine is probably going to break down. You're going to have to replace the column. And in this case, you're going to need to do a lot of batch corrections. So if you run it much faster, this is not only better just for cutting down your costs, but it also usually means your data quality is a little bit higher. So stepping back a little bit, I'll go 10 years ago when shotgun proteomics started to really be used for the first times, or the earliest times, in general biological applications. So if we look back then in the top left, so this shotgun graph, the first problem is that typically these studies, these early studies, might only measure 500 or 1,000 proteins. So this is only 5% of the proteome, and this wouldn't be too bad, but you have to consider that these proteins are generally the most abundant proteins. They're not necessarily in any particular pathway. They're not necessarily interesting to you. The other critical problem beyond this was just the detectable error was really bad. So if you have to run 12 different fractions, that's okay, but if your technical error is 25 or 30% of your measurement, this becomes really a fundamental problem and impossible to do anything particularly fantastic for a biologist with. So of course there's a tremendous need for proteomics, so people continued developing it, and the basis that was provided by shotgun proteomics 10, 15 years ago, this was built upon, and new machines were developed, new informatic approaches were developed. And one of the key ones that came out and is now kind of the gold standard is SRM. So this is a targeted proteomics technique. So in this case, you can only measure much fewer proteins. The time per single measurement is much higher, and you have to develop certain peptides. You have to synthesize these peptides that you actually want to measure to use as a reference. But at least now you can measure hundreds of proteins relatively quickly and with extremely good accuracy. So the most modern techniques like SWATH and the newer developments in shotgun proteomics, these are sort of halfway between the oldest shotgun and the targeted techniques. So now what you might be able to get is you might be able to get 3,000 or 4,000 or even 6,000 proteins if you're doing a large sample run, and now you can get you can get measurement errors more on the scale of 5–10% of the measurement. So there's still room for improvement both in the technical error and in the coverage, but now it's much better than it used to be. And generally, for most people's biological hypotheses, if you can get 4,000 or 6,000 proteins, this is enough to really do pathway analysis for almost anything. It's enough to use de novo hypothesis development to find new proteins to look at specifically, and it's good enough for many people's circumstances today. So that's why mass spectrometry proteomics is now being used more and more commonly with general biologists and non-mass spec specialists such as myself. The last topic I want to bring up here in the introduction is just to remind you what we're actually measuring. So remember, this is bottom-up proteomics, so we're measuring the peptides, not the proteins. So if we look at a relatively average length protein like HAO1 shown here, what we normally do is we digest it with trypsin, which breaks it in at the highlighted arginine and lysine residues. And in the mass spectrometer, we can expect to measure those peptides that are between, say, 6 and 20 amino acids in length. The others would be either too large or too small, or if they're very short, they're not unique to a single protein. And then we have the measurements of all of these peptides that are associated with HAO1 uniquely, and that we think we can use to measure the overall protein levels. So if we actually see it in real data, what we have here is we measured 10 different peptides for HAO1 when we do our SWATH analysis. And in fact, if we do a correlation network, so we do this principal component analysis at the right, what this means is that all of the 9 red samples that are roughly parallel to one another, these are well correlated with one another, whereas the blue sample that's orthogonal, this one is very much non-correlated with the other 9. So in this case, we can say that those 9 peptides that are co-correlated are probably a better correspondence to the overall level of HAO1 than the blue peptide that's orthogonal. This is not necessarily true. It could be that there's a splice variant that we don't know about, but this is identical to how probe set design was done on microarrays. It's identical to how qPCR was done, and the benefit of these newer techniques like SWATH is that you're not actually just measuring 3,648 proteins. You're actually measuring like 40,000 different peptides, which means that for every single protein, you have several different independent assessments that will tell you what the overall level of the protein is. So this gives you a little bit of control on knowing if what you're measuring is what you actually expect it to be. So eventually, as we get more and more developed, we'll actually know which peptides are always good. We'll be able to understand that this blue peptide for HAO1 is bad, and we'll be able to understand why it's bad. But for now, we're still developing these techniques to develop the raw data in the first place. So now I want to move briefly into these multi-omics analyses. So the most common multi-omics analysis people discuss is the transcriptome versus the proteome. So these are fairly easy to layer on one another. You have a gene, and then that gene is transcribed into a transcript, and it's translated into a protein. So it's fairly easy to try and associate these two levels with one another. So in fact, what we've seen in the past 10 or so years since the first proteomics, large-scale proteomics techniques and studies were performed, is we've known that a gene's transcript and protein actually only moderately correlate. But what's interesting is that people give very different responses of how moderately they correlate. Some studies might say 20 or 30% of variance is co-explained, and others might say 70 or 80%. So the difference for this discrepancy, and the large reason for this discrepancy, is that there are two types of correlation people are talking about. So at top, where I say one gene across conditions, this is saying if we perturb a system, so if we give 5 different perturbations, and how much does our gene change at the mRNA level, and how much does it change at the protein level? Do they go in tandem? So in the middle, I show real data for ACSS1, which says that if we perturb the system by looking at different tissues in the same individual, then we see that it's very tightly correlated. The tissues with higher levels of ACSS1 mRNA have much higher levels of protein, and the lower are also lower. But what we can't say is that this is universally true. It's not that ACSS1 is a gene that's universally tightly co-regulated between the 2 layers, and we can trust it, and in your study, you can measure the transcript, and you'll know that the protein is 94% correlated. So here we can take exactly the same data, but rather than all of my tissues, we look just at a subset. So let's take just the heart, and the heart data is shown as a composition of 8 different individuals with different genetic backgrounds. So now I've expanded it out to the 8 different heart individuals with different genetic backgrounds, and now we see the correlation is much weaker. So it's still reasonable. But the reason for this is if you take a look at the axes, and I noticed the axes, I've forgotten its log scale. So if you look on the axes on the middle one, we see that it's about 2 units on the protein in green on the left, and it's about 4 units in mRNA on the right. So this is pretty big differences between the lowest and the highest abundant ACSS1 expressors. If you look at the ACSS1 on the right, where we just look in the heart, we see now it's only 0.4 units on the protein scale, and it's about 0.6 units on the mRNA scale. So this variation is much less within the heart. So the reason that this co-expression is happening much stronger across tissues is that the level of variation is just much higher. So if you have a massive difference in mRNA expression, this is much more likely to have an impact on the protein expression than a small, subtle change. And this is something that's really happening in the cell. It's not due to measurement error. So then the second thing I want to mention is the other type of correlation, which is what's commonly reported if you see people saying that protein and mRNA are 70 or 80% explained by one another. So if instead of looking across tissues, we look across genes within a single sample. So that's saying if within a liver, within a certain liver for an individual, if we just say our machine says these are the highest expressed mRNAs and these are the lowest expressed mRNAs, then if you run that exact same sample on the protein, the higher expressed proteins will be also similar to the higher expressed mRNAs and vice versa. So this is broadly true, but what we're more interested in, actually more likely, is what happens in response to perturbations. If you have cancer, our cells is the expression of certain genes going up or down, and not is it broadly true that these 2 layers of data are fitting with one another. So what this means together is that the large changes are probably consistent. So people who have been doing knockout studies where they saw that their gene that's co-expressed with their knocked out gene is going up or down by 50-fold, these types of problems are almost certainly not going to happen for people who looked at transcriptomics for proteomics. If you have a 50-fold change in the transcript, the protein's almost certainly also going to have a very large change. However, if you're doing population studies, or if you're looking at more subtle factors like for drug testing, if you have a variation of 2 or 3-fold in a transcript level, you can't necessarily take for granted that the protein will also be affected or in the same way. So looking at this a different way, here I want to bring this up one last time. So this is data on the mRNA and protein correlation for 3,700 genes where we've measured both levels of data in several different individuals. So on the x-axis, in the histogram, we show 7 different bins which include all of the mRNAs that are x-fold variable. So I have 68 mRNAs that are more than 2 to the 8-fold variable and 212 that are more than 2 to the 6-fold variable, or 64-fold. So if I look at these most variable transcripts, I can see that these ones that are very highly variable correlate much stronger with their protein. So the average correlation between these very highly variable transcripts, they correlate on average about 0.67 or 0.7 with their protein. So they're much more highly correlated and much more significantly correlated. In the converse sense, if I look at the 299 protein transcripts that are very unvariable, that vary by less than 2-fold across all of my sample types, These ones, on average, correlate only about 0.3 with their corresponding proteins. So of course, this kind of makes sense. If you have a more variable transcript, this will have a more stronger effect on the protein levels. But it's something that people don't necessarily keep in mind when they look at aggregate data of a data set saying, on average, 30% of variation in protein levels can be explained by mRNA. It's true, but what's more interesting is saying, hey, for 500 proteins, 95% of the variation can be explained, and for 200 proteins, none of the variation can be explained. So the way to look at this in a different way is here I've shown on the right. This is TPM1. So on the x-axis, I have the protein. On the y-axis, I have the mRNA. So we see this as a fairly highly variable protein and a fairly highly variable mRNA, and the correlation is very high, although obviously it looks very weird because all the axes are stretched out and there's no intermediary data. I go stepwise. However, the correlation is very good. So this means if we have a big difference in TPM1, we can trust that the mRNA is also going to change. And if we have a small difference, well, now we don't know what's going to happen. Conversely, I can look at a gene like EIF6. So in this case, there's very little variation in the transcript or protein level, maybe even less than twofold, but it's a very linear correlation. Even a small increase in EIF6 mRNA levels will be expressed at the protein level, and you'll see more or less the same thing happen. However, we just know in these ranges we show here, maybe if you made a 50-fold increase in EIF6, maybe the protein would barely go up. So again, we can't say that these are universal truths. So I want to highlight one other thing. Even in cases like EIF6, where I see within this range of expression that is very tightly co-expressed between the mRNA and the transcript, this is for my specific conditions. So maybe in steady state 1, that these are very highly co-expressed and it's reliable and it's correlated, but maybe in an alternative steady state that you're looking at, maybe you don't see the same thing at all and there's no correlation. So essentially what this means is that we're not going to be able to get a database saying, aha, these 5,000 genes, these ones are always very well correlated between the transcript and proteome, which means you don't have to do the proteomics or you don't have to do the transcriptomics. You can do one and say it's an excellent proxy for the other. So this is unlikely to happen, and it's really one of the main reasons that people now need to start looking at proteomics, because now you can do it, which means that there are all sorts of these biological pathways and hypotheses that if you're looking at the transcriptomics before, you just wouldn't have seen it like you would see it today if you did the same thing, the same exact study on the proteomics. So now I want to move into the actual data that shows a couple different examples of how you can use these next-gen proteomics to apply and test completely new hypotheses that were not really possible before, or not at a high throughput at least. So the data I show here, this comes from 40 genetically diverse strains of mouse, BXDs, and we've taken 5 different tissues from each mouse, and then these tissues were processed at the whole tissue level for protein and transcript, and then we did isolated purified mitochondria for the same exact tissues, and then we did the proteomics on these, since naturally there's not much point of doing transcriptomics on just the mitochondria. So looking at this data, because proteomics is very new, you actually need to validate your data pretty much every time you see it. So maybe transcriptomics, you can trust that which proteins you're seeing which transcripts you're seeing on a microarray are correctly defined. In proteins, we're not quite there yet, unless you've already used the exact same system before and already seen these proteins. So the way we can check it one way is we can do hierarchical clustering, and we can see that of these 40 samples I've measured, so 8 strains by each of these 5 tissues, we see that the tissues cluster very distinctly from one another, which is more or less what you'd expect. So this doesn't necessarily mean the data are good, and there are a few resources we can check to make sure that we've actually identified the proteins correctly and that our data match. So we can look at a resource like the Human Protein Atlas, which has been developed over many years, particularly using antibodies, to get the approximate levels of protein expression across tissues. So we can check a few dozen proteins, like here I've shown IMPDH2 and myelin-binding protein, MBP, and we can see that our results are broadly in line with the reported results. So myelin-binding protein, this is brain-specific in the tissues we've measured, whereas IMPDH2 is ubiquitously expressed. We can check a few dozen such, and if they overwhelmingly align with the reported data, we can be sure that our assignments are probably true and that we've correctly assigned all the tissues. So again, this isn't the most interesting thing, but it's something you need to check. What is interesting and what is novel for this new data is we can now look at subcellular localization. So the transcripts don't subcellularly localize particularly well, but the proteins, this is one of these unique studies that we can now do with large-scale proteomics. So before we do that, though, again, we need to also check that our mitochondrial fractions are correctly measured and identified. And the easiest way to do that is probably just to see whether your mitochondria have enrichment of reported mitochondrial proteins. Again, there's a lot of literature, so we can see for our electron transport chain proteins like NDUF-B4 or UQCRB, we can see that these canonical mitochondrial proteins are relatively much higher expressed in the mitochondria than they were in the original fraction. Keep in mind the original fraction does include mitochondria. What's also interesting that we can check is we can look at all of these exceptions. So there are a few dozen proteins, like here I saw ABCC6 and DML5, that were very highly enriched when I did isolated mitochondria, which were in contrast to the reported literature. So again, because this is such a new field, there's a lot more we can discover. So now that we've validated this, we can look into it a little bit more deeply. So here I show the enrichment factor. So essentially this is a histogram showing all of the proteins I've measured, and if a protein is towards farther on the positive x-axis, this means it's higher expressed in mitochondria, and on the left axis means it's lower expressed in mitochondria than the rest of the cell. So if I check this independently for the tissues, I show 3 here. I see that the proteins that are canonical mitochondrial proteins in the red and green, the mitocardia and mitoplast, I see that these are relatively enriched, and I see that proteins that are canonical nucleus or cytosolic, these are unenriched. And I see this uniformly across the 5 tissues, of which I show 3 here. But what's interesting to check is we look at exceptions. It's not black and white. It's not that the green is completely on one side and the blue and gray is completely on the other side. We see there's a fair amount of overlap, although these are z-scores. So if we look at it a little bit more deeply, let's just take the brain here, and let's look, and we see that we have a few of these nuclear proteins, not too many, but a few, that are very highly expressed in the mitochondria, some of which that are even more highly expressed than the average mitochondrial gene. So we can take a look at these, and we can see which of our proteins that are more highly expressed in the mitochondria that we wouldn't expect, which of these are unique to a single tissue, and which of them, like ABCC6, are highly enriched in all 5 of my different tissues, and which of them are sort of in between, like SOT2, which looks like it might be partially expressed in the mitochondria, partially elsewhere, even though SOT2 has never been reported in the mitochondria before. So what we did is we picked a few of these proteins to validate by traditional techniques. So we used the SWATH-MS spectrometry to identify and develop hypotheses, which now we can then go and check in a targeted, more low-throughput technique. So we picked 4 proteins to validate: ABCC6, SOT2, MTAP, and IMPDH, and we identified that they were indeed all mitochondrially associated, even though they were not canonical mitochondrial proteins in the literature. So ABCC6 is in fact part of the mitochondrial-assisted membrane, while the other 3 are actually mitochondrial proteins themselves. So on the Western blots, we see a little bit of difference between these tissues, particularly for IMPDH with its 2 different forms, but we see that these proteins are broadly mitochondrially localized. They are mitochondrial proteins. To be a mitochondrial protein, you don't have to be 100% in the mitochondria. You just have to be there consistently sometimes. So we can look at this a little bit further. So these are Western blots, and what we can also do is we can do immunocytochemistry to try and visualize it. So here we took 2 different cell lines, myotubes and hepatocytes, and we checked 2 of these proteins, MTAP and SOT2. So the red on the TOM20 and the blue on the DAPI, these are the standard antibodies, the standard proteins to blot to tell you where the nucleus is and where the mitochondria is. And this gives you a reference to check against. So then we check MTAP, we check SOT2, and we see, for instance, for MTAP, we see it's actually expressed everywhere. Well, not everywhere, but it's expressed in both the mitochondria and the nucleus in relatively equal proportions, as much as you can distinguish by immunocytochemistry. On the other hand, for SOT2, we see it's primarily in the mitochondria in both of our cell lines, but there is some of it that seems to be in the nucleus. So what's interesting to keep in mind is that this is different from the Western blots I've shown at the upper right. In the Western blots, I see it's massively and predominantly in the mitochondria. There's very little of it in the nuclear and cytosolic fraction, which is called the MC. It's almost all mitochondrial, whereas these cell lines are giving me a much blurrier issue. So this isn't necessarily a problem. It's a different system. You don't expect proteins to always be in the same place all the time without any changes. There are some proteins that are always in the mitochondria or always in the nucleus, but it's not something that's a universal truth. So traditionally, when you're doing antibody techniques, you can't check 50 different conditions on 5 different tissues or 20 different tissues. If you're doing antibody techniques, if you're doing immunocytochemistry, if you're doing Western blots, you really have to pick 1 or 2 things, and you're saying, I'm just going to check HeLa cells and see where proteins are in HeLa, and that's going to be my database. So these have been very useful, these databases, and they're often true, but what we can do with SWATH and these newer mass spectrometry techniques is identify the exceptions, and then we can go in and look at them in further detail, like these 4 that I've mentioned here. So the other example I want to bring up today, and what's uniquely possible with modern proteomics techniques, is looking more functionally. So here we're going to look at the electron transport chain. So the electron transport chain is exclusively part of the mitochondria. All of these proteins are exclusively expressed in the mitochondria, and it plays out in large protein-protein interaction networks. So the electron transport chain, together with the complex 5, forms the oxidative phosphorylation or OXPHOS pathway, sometimes called complex 5. And together, these 5 complexes are composed of about 100 different distinct proteins, which get together in variable conformations. So the textbook example says that you have about 40 proteins that are forming complex 1 always. You have 4 proteins that always make together complex 2. You have like 15 that make complex 3, and so on and so forth. So this is sometimes true, but what research has shown recently is that you don't actually just have these independent complexes. You have these supercomplexes. So instead of having complex 1 shuttling electrons and then going, you have an electron carrier going to complex 3 and then complex 4, when in fact you have these supercomplexes where the complexes are not side by side, but they're actually fit together in some different conformation than if they were side by side. So again, keep in mind this is something we can't do transcriptionally. We have to look at the proteomics of this. So now we can actually try and study this pathway in more detail and see how do these different complexes work and what's the difference between their expression and their makeup in different conditions. So the traditional way people study the entire complexes, you can't do antibody techniques because there are too many different proteins in it. What you can do is a Blue native page or BN page. And in this case, if you isolate the mitochondria, you can just run the isolate on a gel, and you see the oxidative phosphorylation complexes highlighted at different molecular weights. So these are the super complexes, the SC, and they have complex 1 as the largest, complex 5 is the next largest, and so on and so forth. So it can be kind of difficult to distinguish them, but at least you can broadly see what is the expression level of your complexes. So at the right, we can see the same BN page, but now we've highlighted complex 1 and complex 4 with the orange, and the remaining is the blue or purple. But what we also see here is the difference across tissues and the difference across 2 different strains. So DBA is one inbred strain of mouse, and BL6, C57 Black 6, this is another common inbred strain of mouse. So we see not only are there huge differences across the tissues, but we see there are particularly striking differences in genetic background that affect, for instance, supercomplex formation in the liver. And of course, I'm showing you N of 1 and versus N of 1 here, but this holds true if we look at a larger scale. And we will now look at a larger scale. So we had initially seen that there are these differences between Black 6 and DBA mice in their mitochondrial electron transport chain composition. So what we decided to do is we looked at a recombinant inbred family of mice, the BXDs. So the BXDs are a set of about 150 inbred strains of mice that are descended from Black 6 and DBA2, the previous mice I showed in the previous slide. So essentially what we have here is we've phenotyped 40 different strains of inbred mice that are all essentially siblings with one another. They have only 2 possible allelic types, but in different recombinations. So we checked these 46 different sibling strains of mice with 3 biological replicates each, and we see again that there are striking differences in the supercomplex composition, whether they're present or absent in different strains. So here I show 4 different strains, and then here I zoom into just the supercomplexes on all 46 different strains. So again, this is Photoshop spliced to gels, but we're looking at it such a black and white effect that we can see in certain strains like black 6 and 1, we see that there are no upper bands, whereas in DBA and 32 and 40, we see that we do have these upper bands. So this is by BN page, but we can do and see the same thing by mass spec proteomics, and we can try and quantify how much of these proteins there are and their different composition. And what we can do is we can put this together in a QTL map, which is essentially like figuring out what is the genetic cause? Why do half of these strains have these supercomplexes and the other half don't? So we do this QTL map, and we identify one particular locus on chromosome 17 where all of the strains that have the supercomplex have one parental allele, and all the ones that don't have the other parental allele. So we can zoom in on this location on chromosome 17, and we see all of the strains that have the higher supercomplex expression, BXD40, BXD100, DBA2. These have a blue allele, so the DBA2 allele at this location, and all the ones that have lower supercomplex expression or even none, these ones have the red allele at the same location. So this is a QTL map. This is fundamentally the same as a GWAS Manhattan plot. And we can say that there's the causal gene, we're pretty sure that this is a monogenic response because it was so black and white. And we can say that the causal gene is almost certainly within the yellow highlighted box, so this 3-4 megabase region, and it's very likely to even be within the black box, which is about a 1 megabase region. So we can first look at this black box, this 1 megabase region, that conveniently only has 4 possible gene candidates in it. And in fact, of these gene candidates, 3 of them are definitively not in the mitochondria, and 1 of them is, which means the one that is in the mitochondria, COX7A2L, is by far a more likely candidate for affecting supercomplex formation than the other 3 could be. So while we were looking at this, interestingly, while we were developing and phenotyping these mice, in fact, after we'd already done much of it, a paper came out in Science discussing supercomplex assembly and how these supercomplexes are formed and which genes are required. So this paper indicated that COX7A2L is causal, or it's required, for the assembly of certain supercomplexes. So what's interesting is, as we were doing this sort of contemporaneously a little bit behind them, we identified COX7A2L also as certainly required for the development of certain supercomplexes, particularly the ones that I'm highlighting here, these 2 upper bands, bands 5 and 4, which have its complex 1 together with 2 copies of complex 3, together with multiple copies of complex 4. So this is what we saw, and we saw it very clearly, and we see that it aligns very well if we do a Western blot with COX7A2L. We see COX7A2L is there when the bands exist, and when COX7A2L is absent, there are no bands here. So it's not, it's not shut and dry, but it's quite clear to us that COX7A2L is in fact required for these supercomplexes to form. Now, what's interesting is that while we were doing this, a different paper came out saying that no, you've got it all wrong. You absolutely don't need COX7A2L to form these supercomplexes. Maybe it does something, but you definitely don't need it. What was a little bit strange is that this paper that came out that said you don't need it, they actually looked at heart, while the previous study had looked at the liver. So in fact, we checked it ourselves, and we saw that indeed in heart, these same bands that were so exclusive to strains with 1 allele of COX7A2L, these same bands are here. I mean, this isn't very high resolution, but it is true. These same bands, these ones with 2 and 3 copies of complex 4, these exist in the heart, even in the absence of COX7A2L. So what this means is there's some other way to form it in the heart. There's not an exclusive, COX7A2L isn't the exclusive way to form these supercomplexes, but at least in certain conditions and certain tissues it is. And this is as yet unclear quite why. So if we look at it, this in more detail, again, I only showed 2 strains now, but we have done 46. So here I show just 6 different strains. I show 3 that have 1 allele of COX7A2L and 3 that have the misfunctional allele. So between these, if I do my complex activity assays, So this highlights complex 1, 4, 3, etc. And I look at COX7A2L. I can see on the top, here's the liver. On the bottom, here's the heart. So the first thing that's very striking is that the heart is much more complex. There are far more bands in the heart than there are in the liver. And the relative expression is also much higher in the heart than the liver, which is not surprising. Mitochondria are much more abundant in the heart than they are in the liver. But the other thing to keep in mind is, again, when we do this expanded data set, we see very consistently all the time, we see that these upper bands these upper bands highlighted here, these two upper ones are completely absent in all of the liver cases that we see, where all of the animals have the misfunctional allele of COX7A2L. What we also see is that there are a couple other supercomplex bands. We see one that was lower down here that we had previously not seen because it was obscured by complex 5, as it has basically the same molecular weight. But now when we look at this in a different way, we see that again, there's another smaller supercomplex down here that again also requires COX7A2L, at least in the liver. So what's interesting is, again, we look down here at the heart. So this is the same exact animals, same exact time, same everything except a different tissue. And we see again that we see these upper supercomplex bands form even in the absence of COX7A2L. But what's interesting is that we have these lower bands down here, and now instead of one of them, like we have in the liver, now we have two bands in this rough region. One is 2 copies of complex 3 plus 1 copy of complex 4. That's the one I see in the liver. And then the one I see exclusively in the heart is 3 copies of complex 3 plus 2 copies of complex 4. So these ones, even in the heart, it exclusively requires COX7A to form, at least in this data set. So this means that there's some alternative way to form these supercomplexes sometimes, and other times COX7A2L is required. So this is something that you really couldn't have picked up before with transcriptomics. This is now opening a new field. People are looking at supercomplexes much more frequently than they were before because the technology is now possible. It's now possible to measure 100 different proteins reliably across many different samples. And now what we can do is not just say we have one canonical mouse and one canonical tissue. Now we can actually see how all of these different pathways and hypotheses, how these change as a function of different stressors, different tissues, different genetic backgrounds. And this is something where you really need the proteomics. You need these new techniques. The gels I've shown here, they're very nice and visual compared to the mass spectrometry data, unfortunately. But here I can only show you 6 different ones. I can't put 50 different animals together on a single gel. You just wouldn't be able to compare it. Whereas on the other hand, with the mass spectrometry data, this you can actually compare reliably across very much larger sample sets. So that's the gist of the primary talk. I'd just like to thank Yibo Wu and Weishol Wolski for helping with the proteomics, and Pooja Jha, Dongryul, and Junyoung Kim for helping with the mitochondrial isolations and validations, and the direction particularly by Rudy Aebersold, Johanna Auwerks, and then the funding agencies, chiefly the NIH, the ERC, and the Swiss National Science Foundation. So thanks for being here for the talk, and I think we have a few questions that the moderators have highlighted that I will take a look at.
Yes, thank you, Dr. Williams, for that informative presentation. It is now time for Q&A. And I want to remind our audience, if you have a question you'd like to ask Dr. Williams, please do so now. A quick reminder on how to submit them. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We already have some great questions coming in, and we will answer as many of your questions as we have time for. But if we do run out of time, all of your unanswered questions will be answered via email after the presentation. Okay, let's get started. Dr. Williams, our first question is, how can you increase the proteome coverage from about 4,000 you show to 10,000 or the complete proteome?
So the complete proteome is a pretty fundamental problem. There are a lot of proteins that are very highly sequence similar. Like for instance, in the mouse, there are about 1,000 olfactory receptors, and these might only have 1 or 2 different amino acid differences between them, which means they can be very hard or almost impossible to actually measure. You might be able to develop targeted approaches for them, but in any case, the current limit for all of these techniques has seemed to be at about 10,000 proteins. I don't think very many studies have shown beyond that at all. This is somewhat a problem of design of unique peptides, and maybe we'll be able to get to it in the long run. But at least for the moment, the limit is between about 5,000 and 10,000 proteins, at least I think for the next few years.
Okay. Next question. What kind of instrumentation do you need with SWATH?
So you need a mass spectrometer, which is the most fundamental and by far the most expensive. SciEx has a few different machines that work for it. The 5600, the 6600. There are a few benefits to using one or the other, but honestly, as far as I'm aware, there's not a huge difference between them, at least in the way we currently implement it. But of course, it's optimized all the time, so what I say now might not be true in a year. And beyond the mass spectrometer, I mean, you need the LC and the MS, and beyond that, all of the instrumentation is pretty common. You need the centrifuges and all this, but really, it's the LC-MS is the limiting step.
Dr. Williams, does nanotechnology separation help with refining the proteins to be detected?
Yeah, so the development of the libraries, you can do better and better separations, and this does help to some extent, but as you're doing more and more of these, it's actually kind of surprising to me, I don't know quite why, you get very much diminishing returns. So if you do better and better separations, you don't get as much as you expect. So I showed 12 different fractionations before, and if you go to 24 or 36 or 48 fractionations, it surprisingly doesn't really increase your coverage that much. And I think the reason for this might be because really there are a lot of proteins that are highly sequence similar and ribosome or whatever, and these ones are going to be very hard to identify. Maybe there's just no proteotypic peptide that has the correct amino acid length that you expect to see in the mass spectrometer.
Is there still a need for transcriptome measurements once the proteome coverage of samples becomes more complete?
Yeah, so the protein levels of a gene are certainly usually more proximal to its effects on the cell and then overall the cell's action and its phenotypes than the transcripts are. So yeah, if we can measure a protein very reliably, do we need its transcript? So at least right now, we probably do, because we're measuring the level of the protein and not necessarily the activity of the protein. So I think in many cases, you'll probably see perhaps the activity of the protein is better represented by the mRNA level, paradoxically, than looking at the protein level. Maybe the protein is sequestered differently, whereas the mRNA would be actually differentially expressed. So it's hard to say right now. There's not quite enough data on it. And then there's also the non-coding mRNAs, like microarrays, that fundamentally you're always going to need transcriptomics for it.
About population diversity, what happens when you have a study group with lots of genetic variation in protein sequences?
Yeah, so if you have a lot of genetic variation, this has been one of the more fundamental problems. So if you have like 10,000 different humans taken from across the globe, or even stronger if you take mice from different subpopulations or subspecies even, then probably a lot of your measured peptides are going to have allelic variants, which makes A, generating the library very difficult, and B, it can be a little bit tricky to compare levels of two peptides if they're not exactly the same peptide. You can try and do absolute quantification, which helps this a little bit, helps most of it, but then still you need the library to contain all of the possible peptide sequences. And you can use DNA from individuals to try and assume or figure out when you have missense variants and try and put this all together. But fundamentally, it's a bit of a problem. The BXDs, they have 5 million sequence variants, and in fact, we only miss something like 5% of the peptides. And because each protein is represented by 10 or more peptides, we actually don't miss that many proteins in the end. So if you have a lot of genetic variation, it does cut down on the peptides that you have measured. But again, because you have so many peptides per protein, it doesn't have a critical effect. It does degrade your data a little bit, but it's not a massive problem. I think eventually probably you'll be able to integrate it more closely with RNA-seq and DNA data on and know when to expect variants. But right now it's not very good if you're trying to de novo identify missense mutations with mass spectrometry. It's really not developed for that.
Dr. Williams, this is a two-part question. How comprehensive was your SWATH-ION library for mouse? What percentage of the proteome? And will you be making the mouse SWATH library publicly available?
So we used 12 fractionations from 5 different tissues, and we identified about 4,600 proteins in our library. This is, probably a third or a quarter of the overall proteome. And of course, you can extend this by adding in more tissues, running more fractionations, and so forth. And if you look at libraries that have been more definitively developed, like the human SWATH atlas, which I think has all of the NCI 60 cells included, you have a little bit over 10,000 proteins, and some simpler organisms like yeast and tuberculosis have more like 99% of their proteome in the libraries. So it kind of depends what you're looking at. Mouse is surprisingly one of, there's not as much data on the mouse, a bit surprisingly. But yes, it will be publicly available on Proteome Exchange, and the draft library is already there. And they'll also all be available on SWATH Atlas when they're developed.
Now, did you use the same library when processing all the SWATH maps from each of the different organs, or did you have a brain-specific library and a heart-specific library, et cetera?
So we developed the library separately, and then we merged them together and ran all 5 of our tissues using the same library. In our case, this didn't affect our results very much. The fundamental problem is if you have, say, if you're looking at the NCI 60 cell lines, but you're only actually using SWATH on one cell line, then you get a lot of multiple testing and false discovery rate correction, testing for tens of thousands of peptides that aren't even possible to be in your sample. So in our case, we took 5 tissues, and the tissue expression is not, brain is fairly different, but the other 4 are quite common. So it wasn't a big issue for us. You can do it either way. There's no real problem.
Let's see. Next question. Here we go. This is how it's worded. Technical reproducibility of the SWATH-MS measurements relative to the biological variation being measured. This is question number 6.
So for us, the technical error is about 5% of the measurement if we're running technical replicates, sequentially or within the same general approximate time. So that's 5% about error for the technical measurement. The biological error, if we took 2 different animals but from the same genetic background, the same cohort that are siblings in the same cage, these have about 10% error of the measurement. The other problem is technical error across different runs. Like if I run this sample today versus if I run it in 6 months when the mass spec column has been changed and there may be a few other different values, this might be up to 15% of the measurement variance. So if you have very different batches, you have a little bit more of a problem. You can correct this if you know in advance. You can spike in proteins of known quantities like the UPS2 standard and then renormalize your data. But you have to keep in mind in advance whether you want to correct for batches or you think you're going to run everything all at the same time.
We are almost out of time, and this will have to be our last question. But as a reminder, if you did submit a question and we weren't able to get to it, it will be answered via email after the presentation. Dr. Williams, when correlating between metabolites and proteins, do you normally just look at the proteins up and downstream of the metabolite directly, or the metabolite up and downstream of the protein in a pathway, or do you consider more broadly across a pathway?
Yeah, so I mainly talked about the transcriptomics and proteomics today. Multi-omics, using all the omics layers you can possibly measure. This is becoming really popular right now. It's really tricky. Doing DNA to transcript a protein is relatively simple because there's a clear way they're connected to one another. If you look at metabolomics, in this case, people often like looking at flux rather than the steady state levels to see if your perturbation has an effect. If you only have steady state data, as would be the case in like human population studies, then the easiest way is to look for associations directly up or downstream, and you can do metabolite set enrichment analysis, but there has to be a lot of development done right now, to be honest, on integrating metabolomics with the other layers in a way that's consistent across different study pathways. The best way now is certainly to start with hypotheses and check the adjacent proteins. But if you want to de novo find how a metabolite is regulated, this is much harder, and again, you have a lot of problem with multiple testing if you have to check across all of your thousands of proteins to figure out what's modulating your metabolite.
I would like to once again thank Dr. Evan Williams for his presentation. Dr. Williams, do you have any final comments for our audience?
No, so thanks for all of you who attended now, and thanks for all of you who are going to watch this later on the playback on the website. And shoot me an email if you have any questions. I think it directly comes to me through the Q&A button here, although I'm not positive on that. Anyway, thanks for your time.
Thanks again, Dr. Williams. And thank you also to SciEx for making today's educational webinar possible. Now, before we go, I would like to let everyone know that today's webcast will be available for on-demand viewing through June 14th, 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us and hope to see you again soon. Goodbye.